I think we're going to be a bit quicker
Yeah,
now.
we've
And
got
if we do
a few minutes for
if it's too
abnormal
fast for you,
meiosis.
please refer to your handout that you have in front of you. But let's look at abnormal meiosis. Also very important. And this is where an organism always ends up with an incorrect number of chromosomes in their cells, and it's referred to as aneuploidy. So this is, of course, due to mutations that occur during either DNA replication or during transcription of DNA to mRNA, or some external factors like radiation or the effect of heavy metal ions.
Right.
And one of this is, and remember we are busy now here with when meiosis goes wrong, it's Down syndrome. It's mutations that occur. Also referred to as trisomy 21, where
You're not allowed to talk about Mongolism, right?
please, where once again they can give you a karyotype, and they will give you something, and we'll show you now with chromosome pair number 21 will have an extra chromosome. You remember aneuploidy, the wrong number of chromosomes. In this instance, 3 chromosomes instead of 2, and that is why it is trisomy. So there we have it, that the somatic cells ends up with more.
And there you've got
And
a
this
karyotype.
is the karyotype,
Yeah.
and we've highlighted to you that chromosome pair number 21 has an extra chromosome. Please, this is not an extra X chromosome. You can only talk about X and Y chromosomes with chromosome pair number 23. Another mistake that learners
Yeah,
make.
and incidentally, this is a girl now, right?
Yeah.
With, or female, with the 3rd chromosome at pair 21. Okay, Turner syndrome is where there is a problem with the sex chromosomes. And that is where the female has 1 X chromosome missing. And that is because of what they call non-disjunction at chromosome pair 23.
And we'll show them now.
There you've got it, only the 1 X. Klinefelter is where there are 3 an extra sex chromosomes. Then the male has got an extra X. It's also because of non-disjunction at chromosome pair 23. What happened? There you can see Klinefelter, 2X and the Y.
And you know, that's
Yeah.
also the inability of learners to explain themselves what they see. You must remember that you are young scientists, and scientists need to observe, and what you observe you need to write down. So you need to give an evaluation of what you see, and this is what learners can't do. They can't say there's an extra X chromosome or an extra or one less or only 1 single X chromosome on chromosome pair number 23. So make sure that you describe it. Now let's see how this mistake happens. How aneuploidy happens. And this happens, you don't have to know the phases during of meiosis, but just to show you when the homologs, they must pair off at the equator during metaphase. This is what is referred to as non-disjunction. There you see they do not separate, the inability of the homologs or bivalents, they don't separate during meiosis. And what happens if there you can see 3 chromosomes, in this instance goes to the 1 pole and 1 only, so this gamete will end up with 1 less chromosome, and that gamete eventually will have 1 extra chromosome, and this is how non-disjunction can cause aneuploidy.
Yeah, there was a question why always at chromosome 21. It can happen at other chromosomes too. There's a terrible condition that occurs when there's a non-disjunction at chromosome pair 13. There's another terrible condition when it happens at chromosome pair number 18. Chromosome pair 21 is the one that you have to know because Down syndrome is relatively visible, and there are quite sort of a relatively high number of people with Down syndrome living in our community. Some of the other trisomies, non-disjunctions, the condition is so severe that the babies never survive. If they survive until birth, they sometimes die soon after. So yes, it does occur in other chromosomes. The only one that you have to know about is trisomy 21, which is then Down syndrome. And if you are asked to explain how Down syndrome occurs, remember, then we don't ask you the symptoms. Read your questions carefully. If you are asked to explain how Down syndromes occur, you have to say it's abnormal meiosis, non-disjunction, meiosis, And during the anaphase of meiosis 1, anaphase meiosis 1, or you can say anaphase 1, where the chromosome pair does not separate and the one gamete has an extra chromosome 21, and that when this gamete fuses with a normal 23 chromosome gamete, it will result in 47 chromosomes with the extra one, chromosome 21.
Now, if we just quickly look at the next slide, we can see exactly during meiosis 1, the first meiotic division, you can see it's where the homologs, the homologous pairs, they don't separate. It's the non-disjunction happens here. But in meiosis 2, you can see it's where the chromosome, the sister chromatids, they don't separate. So the non-disjunction happens there. Right, we're not going to...
Yeah, that was just an example of a question. And we've had a question from one of the learners with the extra chromosomes in plants and polyploidy that we're going to finish with will explain that.
Yeah, we have polyploidy, and yet is the whole set, the whole diploid set of chromosomes. They don't, it's non-disjunction. And you see, you have to use the word non-disjunction, the inability, they don't separate at all, which means that you will end up with gametes with a diploid number of chromosomes. Some of them even so far as triploid. And as you can see, there's a picture here that where polyploidy happens in plants. It's very rare in animals, of course, but the wheat that we eat today is a result of polyploidy, where the diploid number has been, it's now a hexaploid, where it is 3 times the diploid, the number of chromosomes that we used to have years ago. The characteristics of polyploidy, make sure that you know it, because they are much bigger fruits, bigger flowers. Please make sure not more, but bigger in size. We can create seedless fruits and vegetables, and they're healthier. And with healthier, we mean they're more resistant to diseases. And the one thing, don't, see, this is polyploidy, and see if you can know the difference between polyploidy, make a note of it, and biotechnology, please.
Right, and I think that's all we have time for. Colin, there were some of the learners who said we were going to fast. They cannot take notes. Unfortunately, this is the nature of the presentation, that there isn't time to take notes because we have to deal with a lot of information. But please contact your subject advisor. Regardless of where your school is, contact your subject advisor, because this PowerPoint is available on a handout. And some schools have already received the handout. So yes, you can actually have it on paper so that you don't have to make notes. You can also contact the subject advisor or the departmental representative that will tell you how to contact the telematics division, that will explain to you where you can go and get all the information via the internet. But yes, sorry, cannot take notes, but I'm sure this was just to go through and to help you think of all those important concepts that you have to go and revisit in your own textbook with your teacher.
Yeah, and I think for them, for homework, they can go and look at polygenic inheritance, and so that they can distinguish between polygenic and monogenic inheritance, so that they know exactly. And just to show them the last slide, and maybe we can end off with this, is that here you see it's phenotypes where there's continuous spectrum, and we call it continuous variation. If you look at your mother and you look at your father and you look at all your brothers and sisters and yourself, you will see that all of you have different heights, and this is because we refer to this, and if we go back, it is governed by more than one gene pair on different loci. So here we have a lot of intermediate phenotypes for this, and this is what is referred to as continuous or a variation.
I think it's the same with skin color and hair color, and yes, and there you can see actually a beautiful photo.
Yeah, some of them will identify the very important, the stars there, but here you can see father and the mother and continuous variation, that you can see all of them different skin colors. And this is then polygenic inheritance, and we end off today's session with them.
It was a marathon session.
It was.
And yeah, we see you soon. and have a good week and work hard. The exams are upon you. And remember, we're working towards November, where we expect excellent results of every hard worker.
And the challenge, of course, is to go home now and go through this work again, because one of these days is exam time. So from us, goodbye.
Keep well.